Today, we will be covering the topic the gram-positive bacteria. But before that, we'll see the general characteristics of bacteria. So these bacteria are prokaryotic, microscopic, single-cell organisms that thrive in a diverse environment and constitute a large domain of prokaryotic organisms. They can live within the soil, in ocean, and inside the human gut. The size of the bacteria, it ranges from 0.5 to 1 micrometer, and the shape of the bacteria is governed by their rigid cell wall. So this bacteria possess a cell wall that is outer to the cytoplasmic membrane. The cell wall is a very rigid structure that gives shape to the cell, and their function is to prevent the cell from expanding and bursting because of the uptake of water. As most bacteria, they thrive in the hypotonic environment. Cell wall is usually essential for bacterial growth and division. Bacterial cell wall possesses peptidoglycan and is chemically and morphologically very complex. A typical bacterial cell may be spherical, which we call it as a cocci, or straight rods, also known as bacilli, or rods that are helically curved. But some bacteria may be pleomorphic, that is, they can exhibit a variety of shapes. Bacteria are classified as gram-positive or gram-negative, and it is based on a differential staining properties which is known as the gram staining technique. This gram staining technique was developed by Dr. Hans Christian Gram in the year 1884. In this technique, the Gram reaction is related to the permeability of the bacterial cell wall and cytoplasmic membrane to the diiodine complex. The gram-positive cells restrict the outflow of the complex during decolorization, whereas in the case of gram-negative wall, it allows it. So next we'll see the mechanism of gram staining. In gram staining technique, the primary stain is the crystal violet stain. The microorganism or the bacteria that retain the crystal violet iodine complex appear purple under the microscope. Such organisms are commonly classified as gram-positive, whereas others that do not stain crystal violet are the gram-negative organism. Here, gram staining is based primarily on the ability of the bacterial cell wall to retain the crystal violet dye. The cell wall of gram-positive organism, they have a higher peptidoglycan and lower lipid content than the gram-negative organism. So after the cells that are treated with crystal violet, they are subsequently treated with iodine to promote the dye retention. However, subsequent treatment with a decolorizer, it dissolves the lipid layer from the gram-negative cells. And this removal of the lipid layer enhances the leaching of the primary stain from the cells into the surrounding solvent. In contrast, in gram-positive bacteria, the solvent dehydrates the thicker gram-positive cell wall and closes the pores as the cell wall shrinks during dehydration. And as a result, the diffusion of the crystal violet iodine complex is blocked and the bacteria remains stained, that is the purple color. Finally, a counter stain that is known as safranin is applied to the smear to give decolorized gram-negative bacteria a pink color. Next, we'll come to the structure of peptidoglycan. So now we come to know that the cell wall of both gram-positive and gram-negative bacteria possess peptidoglycan, and the only difference is that the gram-positive have higher content of peptidoglycan than the gram-negative bacteria. Here, peptidoglycan is also known as murin, and it is a polymer containing two sugar derivatives, that is N-acetylglucosamine and N-acetylmuramic acid, and many different amino acids. This N-acetylmuramic acid is the lactyl ether of N-acetylglucosamine, and the lactate group of the muramic acid forms an amide link with the amino terminus of a short peptide containing 4 to 6 amino acid residues. Further, the peptide extension can form cross bridge connecting these parallel strands of glycans. D-glutamic acid, D-alanine, and mesodiaminopamelic acids are some of the amino acids that are present in the peptidoglycan. And the backbone of the peptidoglycan is composed of alternating N-acetylglucosamine and N-acetylmuramic acid residues, and a peptide chain of 4 alternating D and L amino acid is connected to the carboxyl group of N-acetylmuramic acid. Further, the carboxyl group of the terminal D-alanine is connected to the amino group of diaminopamelic acid. The cross-linking between amino acid in different linear amino sugars chain occur with the help of an enzyme that is known as transpeptidase, and as a result, it forms a strong and a rigid three-dimensional structure. But the specific amino acid sequence and the molecular structure, it varies with the bacterial species. So when we take an example of some gram-positive bacterial species such as Staphylococcus aureus, which is responsible for toxic shock syndrome, have peptides that are linked by bridges of pentaglycine instead of the D-alanine that is linked to diaminopamelic acid. Peptidoglycan gives structural strength to the bacterial cells, and also it maintains the osmotic pressure of the cell cytoplasm. Next, we'll come to the gram-positive cell wall. Although the thick homogeneous cell wall of gram-positive bacteria is composed primarily of peptidoglycan, it also possesses a large amount of teichoic acid. So this teichoic acid is a polymer of glycerol or ribitol that is joined by a phosphate groups. And to this glycerol and ribitol groups, amino acids such as D-alanine or sugars like glucose are attached. Further, teichoic acids are attached to the peptidoglycan with 6-hydroxyl of N-acetylmuramic acid by a covalent bond or to the plasma membrane lipids. When these teichoic acids are attached to the plasma membrane lipids, they are termed as lipoteichoic acid. Now, when we talk about the charge that is carried by the gram-positive cell wall, they possess a negative charge. This negative charge is conferred by the extension of the teichoic acid, which is negatively charged on the surface of the peptidoglycans. This teichoic acid is assumed to play a very important role in maintaining the structure of the cell wall, but their exact function still remains unclear. Further, the cell wall of gram-positive bacteria, it contains a very little amount of lipid, but Mycobacterium, Corynebacterium, and certain other genera are rich in lipid, and hence the ability of mycobacteria to exhibit acid-fast staining, which is related to the presence of mycolic acid in their cell wall. So next we'll see the gram-positive envelope. From the above discussion, we come to know that gram-positive bacterial cell wall consists of multiple layers of peptidoglycan and is reinforced by the teichoic acid. Also, the gram-positive cell wall is enclosed by a slippery capsule consisting of loosely bound polysaccharides. And these capsules are not required for bacterial growth and reproduction. They help bacteria resist phagocytosis by host phagocytic cells. Capsules also contain a great deal of water and protect bacteria against desiccation. Bacterial viruses and most hydrophobic toxic material such as detergents are excluded by capsules. Capsules also facilitate the attachment of bacteria on surface of solid objects in aquatic environment or to tissue surfaces in plants and animal hosts. They also possess, that is, the gram-positive bacteria also possess an additional protective layer that is known as ACE layer or the surface layer. This ACE layer is a crystalline layer which is composed of proteins or glycoproteins. And the glycan chain of ACE layer proteins are similar to the lipopolysaccharides occurring in the outer membrane of gram-negative bacteria. Each subunit of S layer contains a pore to admit a wide range of molecules. And these subunits forms a smooth layer on the cell wall or outer membrane, and the proteins are arranged either hexagonally or tetragonally. The S layer also forms a rigid layer, but sometimes it also flexes and allows certain substances to pass through it. And when we come to the function of S layer is that it may protect the cell against ion and pH fluctuation, osmotic stress, enzymes, etc. They also helps to maintain the shape and envelope rigidity of some bacterial cell. S layer also seems to protect some pathogens against complement attack and phagocytosis, and these contribute to their virulence. Next, we'll see the gram-positive bacteria. Now, we all know that gram-positive bacteria have a thick cell wall with multiple layers of peptidoglycan interpenetrated by teichoic acid. And these gram-positive species make up the phylum Firmicutes. And most of the gram-positive bacteria have a very simple morphology. Some of the genera have the capacity to form a heat-resistant forms that is known as endospore. And these endospore are formed intracellularly and hence the name, that is endospore. They are highly resistant to heat, ultraviolet and ionizing radiation and many toxic chemicals. But a number of gram-positive bacteria are asporogenous and are anaerobic or facultatively anaerobic organism. And some of the gram-positive bacteria are filamentous and develop a mycelial growth habitat. And when we come to motility in gram-positive organisms are carried out by means of a flagella. And in case of reproduction, the mode of reproduction is by means of transverse binary fission. Some of the gram-positive bacteria includes Staphylococcus aureus, Streptococcus mutans, Bacillus anthracis, Clostridium tetani, Corynebacterium diphtheria, and so on. The gram-positive bacteria that is included in the Burgess Manual Volume 2 includes, the first one is gram-positive cocci, The organism under this may have a strict respiratory type of metabolism, a respiratory and a fermentative metabolism, or a strictly fermentative metabolism. And these bacteria that undergo strict fermentative metabolism may grow in the presence of air, that is aerotolerant or maybe anaerobic. The second one is endospore forming gram-positive bacteria. Organisms under this are mostly rod-shaped, but some are cocci. They range from aerobic to facultative anaerobic to anaerobic. And majority of the anaerobes, they survive by fermentation but also respire anaerobically with sulfur. The third one, non-spore forming gram-positive rods of regular shape. The cells of the organism under this group have a uniform appearance without swelling or branching. They are aerobes, facultative anaerobes, or aerotolerant anaerobes. The fourth point is non-spore forming gram-positive rods of irregular shape. The cells of this organism exhibit swelling. The shapes of the bacteria also appear Y or V shape, rods or coccus, or they do not form or have a uniform morphology. They are aerobic and facultative anaerobic organism. Next point, mycobacteria. Mycobacteria are aerobic and are slightly curved or a straight rods and sometimes shows branching. These group of bacteria are acid-fast organisms because of the presence of mycolic acid. This last one, nocardioforms. Nocardioforms are aerobic organisms that tend to form substrate mycelium and aerial mycelium. Conidiospores may develop from the aerial hypha. Further, the hypha may fragment into rod-shaped or coccoid shape. So, from the above discussion, we can conclude that the classification of bacteria as gram-positive and gram-negative is based on the differences in the cell wall structure and their response to gram staining. Gram-positive cell walls have thick homogeneous layer of peptidoglycans and teichoic acid, and 90% of peptidoglycans make up the cell wall. This layer forms the outermost structure of the gram-positive cells and confers strength to the cell wall. Finally, the chemical complexity of the bacterial cell wall is due to the content of the peptidoglycans.